I am Meghna Janardhanan here before you to introduce and host the next session. So I am a final year integrated master student in molecular biology at University of Mysore. I am also the chief of marketing and capacity building at Project Encephalon. Up next, we have an invited talk by Dr. Madhurima Benekareddy on drug discovery in neuroscience and rare diseases. Dr. Madhurima is currently a senior scientist, research project leader at Roche, Switzerland, network head, movement disorder ad interim. Having completed her bachelor's in science at Osmania University, Hyderabad, she obtained her PhD from Tata Institute of Fundamental Research, Mumbai, following which she worked as a postdoctoral fellow at Roche, Switzerland, where she later served as scientist and lab head. She has extensive research experience in leading preclinical research aimed at elucidating disease biology and mechanistic understanding in neuroscience and rare diseases using in vitro and in vivo approaches. She is also leading the matrix function of movement disorders ad interim with oversight on strategy and portfolio for Huntington's disease, Parkinson's disease, and ataxis. She has also been actively contributing to advanced stage drug discovery projects and designing in context to in vivo fMRI and calcium imaging. She is interested in furthering creative approaches to drug discovery in neuroscience and rare diseases and nurturing a creative and inclusive people culture. We are elated to have you here and look forward to your talk. Over to you, Dr. Mathurima.
Thank you, Meghna. I'm really happy to attend the convention and really enjoyed Poonam's talk before. Obviously, it's something very close to my heart. So I'm really excited to talk to you about some of the work that I've been doing over the past 9 years now at Roche. And I'm going to try to, can I share the screen? It says it's going to, I think, okay. So let's see how this works. All right, just bear with me while I put this in presentation mode. Okay, hope this works now. Yeah, okay, good. So yeah, I'm very happy to talk to you about some of my perspectives on drug discovery in neuroscience and rare diseases based on the work that I've been doing at Roche over the past 9 years. So before I kind of dive in, the slide is just to remind me to talk a little bit about Roche. Having interacted at conferences, I feel like this is something that there is not a lot of visibility to in general about drug discovery in pharma. And so maybe I thought I'll spend a couple of minutes talking briefly about it, just based on my own career. So I did my PhD at TIFR with Vidita, and then after that I was interested, as you know, Vidita's lab works on stress and depression, and that really sparked my interest in understanding disease biology and contributing to therapeutics. And so I went to Roche for a postdoc, did my postdoc with Dr. Anurwan Ghosh, and there the idea of the postdoc was trying to understand a little bit about how can we use some of the systems neuroscience tools to develop models for social behavior, to understand social behavior in a normal condition as well as in a disease state, and can we use therapies that we're developing at Roche to see, to test in these models and understand whether you can use this as a disease model. And then after that, so after my postdoc, we published some of the work that we did. And then I stayed on as a scientist to apply some of these models in drug discovery. So then started really using these models, testing drugs that are in the pipeline to understand, you know, how do these work? Do they work in a way that would be expected? And based on how they work in these models, can you say something about their effect on behavior and kind of their use in a disease condition? And that was really interesting. And then after that, one of the really cool things about being in a neuroscience, in a place like Roche, we have a big neuroscience and rare disease department. And there is a lot of interest broadly in different areas of neuroscience. And I've been very, as you know, I really liked the kind of really broad neuroscience experience at TIFR, and I was really generally curious about what other neuroscience stuff's going on at Roche, and this kind of led me to working on Huntington's disease. And about a year and a half, I transitioned into working on Huntington's disease full-time, and there the focus is slightly different. I'll talk to you a little bit about it. There I'm working more on kind of platforms and technologies of how to target this gene. So this is kind of overall how I've kind of over the past few years transitioned from different areas of drug discovery neuroscience and using different methodologies. So with that, this is just an overview of today's talk. I'm going to talk about two different approaches for developing pro-social therapies. So two different approaches to drug discovery. One of them is you have a kind of a large indication like autism spectrum disorder, or you have kind of a rare disease like Huntington's disease, and there's kind of diverse approaches to drug discovery in these two areas. The first one is, so when you talk about like an area like ASD, I was working on developing pro-social therapies, and there is kind of two main areas that I worked on, kind of understanding the biology underlying the circuitry of social behavior. And then one could think of using these circuits and finding specific targets like a GPCR or another molecular pathway that is specific to this circuit, and using that as a target for a therapeutic. And we did some of this work. I'm not going to talk about this in the interest of time, but we're working on a manuscript, so stay tuned. What I'll talk about is how we worked on this to develop a circuit model for social disruptions, and how we use that to test some pro-social therapies. And then I'll talk a little bit about Huntington's disease, where the focus is on technologies, because obviously we already know the target. We have genetic models, and there the challenge is now, how do you make specific therapies for lowering Huntington's disease? And I'll talk a little bit about some of kind of what is in the field, and then some of the work that I have done in this area. So to start with, I'll start with my ASD work, kind of starting chronologically about using circuit models for developing therapeutics. So I guess as some of you who work in systems neuroscience may be familiar, starting from kind of Yizhar's work when he was in Carl Weissert's lab, there was a lot of interest in prefrontal cortex as a starting point for analyzing social behavior. Certainly, I have been really inspired by Yizhar's original paper. And when I started my postdoc, I think that was just, that paper had just come out, and we had thought about like, so this is a good kind of starting point, because if you look at any fMRI data or other kind of circuit-based data where people with social disruptions have been, like when you look at brains, there's always this kind of aspect of prefrontal cortex. And if you look at, for instance, from this review, you can see that the prefrontal cortex, different areas of the prefrontal cortex are often shown to be impaired in many disease areas like schizophrenia, autism, and depression, where there is social disruptions. In addition, what is of interest, I'm not going to talk about this, but this is stuff that I've worked on and it's close to my heart, is other areas that also are thought to be part of this kind of social brain network, right? Things like the habenula, anterior cingulate cortex, amygdala, accumbens, VTA, and so on. So there is a lot of circuitry, and of course, it is. I have to say a disclaimer right away that my approach is certainly very reductionist, and in terms of actual systems neuroscience and really understanding how the brain produces social behavior, that is certainly very complex. But I was trying to capture kind of one of like a fundamental concepts and trying to use that for predicting how therapeutics would work. And this is actually one of the big differences of how if I was in academia, I would probably be thinking more about exactly how the brain produces social behavior. But I'm actually interested in how can you make a therapeutic if there is a social behavior disruption. So I kind of use one of the general principles to make a model. And so how did I do that? So, like I said, we started with expression with prefrontal cortex, and we used chemogenetic tools from developed in Brian Roth's lab. Some of this early work has been published, where basically we said, let's kind of start with the basic idea that you would disrupt prefrontal cortex activity. We've done both kind of acute, chronic, increasing activity, decreasing activity with different cell types. But kind of one of the general principles that shows up and is in line with Yi's theory is that if you kind of disrupt the activity, information processing, information flow in the prefrontal cortex, that would be detrimental for social behavior, just because this is such a key area for social information processing. So that's kind of a broad model that I've used. And we've been able to reproduce some of the data where prefrontal cortex disruption of the activity leads to deficits in social behavior. And the way we measured social behavior was using this 3-chamber sociability assay. So as you can see here, we kind of have 10 days of administration. Inject HM3D into the prefrontal cortex, administer the ligand CNO for 10 days, and then we look at social behavior at the beginning and at the end, but we also look at it after, so after 10 days of administration, we have a recovery period of 1 week to not to have any acute effects of any of the drugs on board, but just to look at chronic effects on the circuit. So what you're looking at here is day 1 and day 18, and you can kind of see that there is, when you have just vehicle, there's obviously a lot of test-retest variability, but when you have a circuitry that has been disrupted, where the prefrontal activity has been disrupted, we see that the sociability index goes down quite a bit. And I think one of the main questions I was interested in is, can a pro-social therapy be used to prevent this disruption of prefrontal cortex activity and social behavior? And I think this is the paradigm that I explained before. So we did social behavior baseline. So these all of the rats received a surgical infusion of HM3D in the prefrontal cortex, and then there, the usual 3-week recovery period, then measured sociability at baseline, and then we had daily administration of CNO to increase activity in the prefrontal cortex. Then after day 10, we have kind of this recovery period during which we either gave the vehicle or a pro-social therapy. So this is the idea, is that in this kind of time, can you prevent, after this insult, can you prevent disruption of social behavior? And what we saw was that, and we used here an oxytocin agonist. This is something that is different from natural oxytocin, but this is an oxytocin agonist that is developed by Roche. And so what you see here is that we use 3 different doses of this oxytocin receptor agonist. And you can see here is control, and with prefrontal cortex activation, what I'm measuring here is sniffing time, so the interaction time in a 3-chamber sociability task that I showed before. And you see that the social interaction time goes down with this chronic PFC activation. And then you with increasing doses of the oxytocin receptor agonist, you see this reversal of social behavior. And then, of course, we did a lot of work, and we are working on a manuscript here. So I just want to give a flavor and kind of stay tuned for some of the work that we're going to publish shortly. But I just want to leave you with this kind of take-home message that treatment with oxytocin receptor agonist indeed reverses the change in social behavior. And what this gives you is a model for understanding social deficits. But of course, we were interested on the side on what are the mechanisms that's going on. Obviously, this is a good model to say test therapies, but what is kind of what's going on in the molecular mechanisms. So this is with a talented intern at Roche. We were able to work on this. And I'm not a physiologist, so I'm not presenting a lot of physiology, but there is so we did a lot to understand what happens at the level of the circuitry. And so this is again the experimental groups that I discussed so far. So all of the rats received HM3D infusions. Then you have a 2 to 3 week recovery period, CNO infusions, and then you have either vehicle, oxytocin agonist, or then you have where there is a M-Sherry and CNO control, they still receive vehicle. And what Philip saw when he looked at this was he looked at intrinsic excitability, and this is one of the measures. He did a lot of work, and in the prefrontal cortex, the intrinsic excitability was higher. So it was basically intrinsic excitability was lower, and it was reversed by oxytocin treatment. And so you may be confused a little bit because the model itself is reversal, is prefrontal cortex activation, but it turns out that when you have this kind of chronic activation of the prefrontal cortex, you decrease intrinsic excitability, and that makes sense because obviously the in terms of scaling and homeostatic kind of changes that happen in the brain. So that's why kind of it goes back to one of the first comment I was making was basically to say that the idea that you either increase or decrease the prefrontal cortex activity but just disrupt it, that seems to be the kind of common denominator to models that lead to disruption of social behavior. And so we did a little more characterization here. I don't have time to get into it, but this is some of the ways in which one can use circuit models to test therapeutics, obviously, but also to understand the molecular mechanisms and perhaps discover new targets where you could target a certain pathway and cause prosocial effects and kind of form a basis for prosocial therapies. So I just want to end this the circuit part and move to the next part. So what I kind of wanted to I hope I was able to impress upon you that the general concepts of social using kind of circuit neuroscience was I kind of tested some basic principles. One of them is that when I disrupt prefrontal cortex activity using various paradigms, I'm able to decrease sociability. And in some of the work that we published previously, we were also able to show using an fMRI that we can modulate relevant circuitry. And this concept is really important because this establishes a framework for using these circuit-based manipulations combined with pharmacological interventions like oxytocin receptor agonists that I showed to correct social deficits. And this kind of knowledge about the circuit concepts, this informs ongoing efforts that we have at developing targeting therapeutics. And so this is basically the way we think about it. And with that, I'd like to move to my kind of second concept. So far I talked about using circuit models for developing prosocial therapies, where here there is kind of a distributed genetic landscape. So often the target biology is not clear. So there are some rare genetic variants, but there's also pathway targets, circuit targets. And here the disease models are both genetic circuit models. And here I spoke a little bit about discovery of circuit-based targets, because here the focus very much is on new target discovery and also using circuit models for developing therapeutics. And then what I want to talk to you in the next 15 minutes or so is about a different area that I'm working on currently, which is Huntington's disease. Huntington's disease is a monogenetic disease caused by triplet repeat expansion in the exon 1 of the gene. It leads to gain of toxic function. And because you know the cause of the disease, the target biology is clear, right? You have Huntington you have to lower Huntington because it leads to gain of toxic function. And the disease models are there because, of course, when you know the cause of the disease, it's easy to have a genetic model, or it's easy to have patient-derived iPSC neurons to model the disease in vitro. And so where's the focus for us is to technologies and platforms to target Huntington. So I'm going to talk to you a little bit about two of the stuff. So antisense oligos, this is basically a review of what is the status of the field and some of the work that we have done on early gene editing technologies that we've done. So this is kind of a little overview of Huntington's disease and how we think about Huntington lowering therapies in this area. So this is a mode of action of kind of what is the pathology at the cellular level. As you can see, this is a busy slide, but on top here, so you have the expansion, there is pre-mRNA and mRNA. And what I'm going to talk about initially is therapies kind of close to proximal to the cause of the disease. So I'll first talk about antisense oligonucleotide, which is kind of like a synthetic DNA, if you will, that binds through Watson group base pairing to pre-mRNA, RNA and pre-mRNA. and then leads to recruitment of this enzyme called RNase H, and that leads to degradation of mRNA and pre-mRNA. And this leads to lowering of Huntington protein. So this, as you can imagine, lowering here would kind of arrest all the toxic pathology, pathological causes of the disease, toxic mutant Huntington protein aggregation that leads to impaired axonal transport, neuronal death, excitotoxicity, and at a circuit level, leading to neurodegeneration, atrophy, cognitive behavioral dysfunction, as well as motor dysfunction. And so I'll talk to you a little bit about what has been done in the field, in the ASO field, and then some of the work that we're doing to enable even more advanced therapies that could be proximal to the cause of the disease. So like I mentioned, direct gene targeting and innovative platforms are how we think about therapies for Huntington's disease. A quick reminder again, antisense oligonucleotides are synthetic drugs. So it's basically synthetic DNA-based drugs. And these are modified DNA drugs. And these are designed to be complementary to specific regions in the pre-mRNA or mRNA of Huntington. And upon binding with Watson-Crick base pairing to this target, they lead to the recruitment of RNase H and mediated degradation. This leads to decrease in the mRNA and then decrease in the protein. So as I mentioned before, the target biology is clear because we believe in that Huntington lowering therapies would be transformational. We do have very clear genetic models. I'll talk to you a little bit about it. There are many models where you kind of have a transgene of the human mutant Huntington gene, and that leads to that leads to replication of the etiology of Huntington's disease. And I'll talk about ASOs and gene editing here. So this is a busy slide, and this is work, seminal work done by Holly Kotasevich, who's now at Ionis, and this has been published. This has been published for a while now, but this is basically behind one of the antisense oligo drugs, which is in phase 3 in the clinic. So on the left is kind of modulation of the target in vivo, and on the right is behavior. So these are all these are all experiments done in packaged D mouse. So like I mentioned, we have a mouse model, transgenic mouse model of Huntington's disease, where there is this antisense oligo has been injected. And you see that with there's a dose-dependent decrease in levels of Huntington mRNA with this antisense oligonucleotide. And as you can see that this leads, so there's there's a dose-dependent increase, but there's also time-dependent increase. And so this increase lasts for up to 12 weeks. And even at 16 weeks, this kind of comes back only to about 75%. So this is also long-lasting after a single administration. And this is, if you look at this, the concentration, this is actually the concentration of the drug in the tissue. This is very interesting because at 12 months, the amount of drug in the tissue is very low, but it looks like it looks like it's able to come back. It's able to suppress the amount of drug, suppress the amount of Huntington mRNA, despite having very low concentrations of the drug at that time point. And this kind of points to one of the interesting things in the ASO field is that once you kind of get enough drug into the active compartment, you're probably able to, active compartment meaning a specific compartment in the cell, perhaps the nucleus in this case, you're able to cause significant lowering because these drugs are very stable. And these are the level of mRNA, and this is at the level of the protein. And you see that there is there is a lag in the effect of the mRNA and the protein, but still both the effect on mRNA and protein are long lasting. So what about behavior, right? So in the same mouse model, Holly did rotor-rod experiments, and rotor-rod is this kind of rotating kind of axis. There's a rotating wheel, if you will, but the mice, instead of running inside the wheel, they kind of run on the outside of the wheel. This is kind of a simplified explanation of the rotor-rod. And mice usually like to run, and it's kind of like a nice game for them. But as you accelerate the speed, at some point they can't keep up and they kind of jump off the rod. And that's basically what I'm showing here is what is the latency to fall off the rod. And as you can see with the non-transgenic mice, the test, so we test them for about like 5 minutes, that's 300 seconds, and they kind of play around with it for until about 200 seconds on an average. Whereas if you have a BAC-HT, which is a Huntington's disease transgenic mice, you see that they're already at the 6-week time point, they have a significant pathology. This is because they have 4 copies of this transgene. So the kind of progression of the disease is obviously much faster here in the mice. And so there's already at baseline a decrease in the latency. However, on treatment with azos, you can see that there is a recovery of this behavior. And this recovery is long lasting. It doesn't quite get back to normal, but it seems like that it's significantly changed from without treatment. So this is kind of, there's a lot of work done on how this works and so on. So this is a very interesting field. It's certainly a very active for scientific investigation. But I just wanted to talk to you about it just to give you the idea of what is the science behind this drug, because now this drug is in phase 3. And what do we know from patients, right? How much of this translates? And that is the really interesting thing about being in a field where you have a drug that's in the clinic. You can really learn from what's in the clinic, but there's also, like Poonam mentioned, a huge potential for biotranslation. And I think that is also one of the things that I'm really interested in terms of using some of the information we have from the clinic to make meta models to enable us to develop better second generation therapies. And so, as you can see, so this tominersen is what the AZO is called. Is given to patients with Huntington's disease, and then you see that you can measure the levels of mutant Huntington in the CSF. Obviously, you can't biopsy the brain, so you have to work with what you have. But luckily, you can measure this biomarker mutant Huntington in the CSF, and as you can see, there is a dose-dependent decrease in the amounts of mutant Huntington in the CSF upon administration of this drug. And here you can also see kind of a time-dependent effect. This is a monthly administration, 4 administrations, and even after the administration is stopped, there seems to be a decrease in Huntington protein, which is reminiscent a little bit which is in line with our understanding from in vivo studies that this is it's a very long-lasting and stable drug. And this is work done in collaboration with Ionis, who's our long-term partner now for the tominersen development. And with this, so I want to leave you with this on the AZO front, just in the interest of time, and move on to the next modality for Huntington lowering that I want to talk about. And this is gene editing. So why do we think that we need a second generation technology? Right? So obviously AZO therapeutics are great, but we are interested in kind of a one-time administration, and also an allele-specific lowering of Huntington's disease. And that is possible with gene editing. Of course, this is not currently viable. Gene editing is in the clinic, let's say, for things like TTR for peripheral diseases, but gene editing for CNS is currently still in stages of early development. And so what is the main challenge here, right? The main challenge here is actually delivery. So as you know, CRISPR-Cas9 based editing the concept is basically that you have Cas9 nuclease, which uses the PAM to bind to the target area on the genome. And then you have guide RNA that gives additional specificity. And then that leads to double-stranded DNA breaks and production of indels and inactivation of the gene and consequent lowering of the gene product. And so that is the idea. And I'm not going to talk to you about this because this is actually work that we are in the process of submitting to a manuscript. And I just want to talk to you a little bit about some of the work that we did to enable prior to this, is how do we deliver this? But of course, so there's 2 aspects. You want to understand how to edit the gene itself, but another major question is how do you deliver this nuclease and along with this guide RNA, because this is Cas9 is kind of like the size of an antibody. If you just inject it into your blood, it's not going to go to the brain. It's not going to cross the blood-brain barrier. So how do we solve this problem? And so people have thought about this quite a bit, and this is work we've done in collaboration with Jennifer Doudna's lab when I was a postdoc, and now we're continuing that. So people have thought about this many ways. Right? One can have like a genetically encoded kind of delivery, so you can have a plasmid DNA, a viral vector, or you can have an mRNA drug with an LNP. This is kind of what the Pfizer and the COVID vaccine would be like, is basically mRNA and encapsulated in an LNP. And then there is also non-genetically encoded, where you just have an RNP, which is basically, if you think about it, is like enzyme therapy, right? So you make the Cas9 enzyme and then you deliver it locally. And that is basically what is called RNP, mode of delivery, ribonuclear protein. So this is non-genetically encoded. It has a lot of advantages and disadvantages. And the main advantage of non-viral delivery, either LNP or RNP, is that you would not have the genetic information in the cell, and that would be and since you have kind of like a one-time delivery, you just want kind of a hit and go kind of method, right? So you want the gene editing, and then once, and then you don't want it anymore, because once the job's done, you don't need the drug substance to be present in your cells all the time. That could lead to some consequences that you don't want. And that's the reason a lot of people really like non-viral delivery. And so we wanted to understand whether this can be feasible using a reporter system. And what we did was use the Th tomato mice. And as you know, there's a Th tomato insertion in the rosal locus, and the expression of this Th tomato is blocked because of a stop cassette. And you can have and this is flanked by LOXP sites. And usually we have all used this, you inject like a Cree virus or something where you can kind of remove the stop cassette or invert it, so you can have expression of Th tomato. What we did here was to use the same system, but for Cas9. And one of the ideas here was to design the guide RNA so you can have double-stranded breaks in the stop cassette, leading to kind of inactivation of the stop cassette, and then that leads to expression of Th tomato. And that's kind of what I tried to show here. So we use the system, and there is two components, right? One component is like to make the Cas9, and the other component is the cells. So we tested them on NPCs from these Th tomato mice. And what Brett did is made the Cas9, and then you have the guide RNA using in vitro transcription. You kind of load the guide RNA onto Cas9, and then lead to and Cas9, and then treat the cells, or you can treat the mouse. So what I'm going to show you is some of the work that we did in my lab on this mouse model. So a lot of the in vitro work was done at Berkeley, and we did the in vivo work at Roche. And what did the in vivo work involve is taking the native Cas9, protein engineering it, and we call it neuron penetrating Cas9, lowering the guide RNA, and then surgical infusion into the brain, into the striatum, bilateral, and you can see expression. If there is genome editing, you will see expression of Th tomato in red. So what kind of protein engineering did Brett do? So basically, Brett kind of played around with nuclear localization signals at the C terminus and N terminus, and it turns out that the 4x NLS seems to be the one that increased the efficacy in vitro, so in the NPC model. And then so basically we went ahead with this 4x NLS version in mouse studies. And as I mentioned before, we did bilateral infusion of this in Th tomato mice. And you can see that if you make a really focused injection, that you would see red cells. And it's very easy to screen the red cells. And then we were also able to estimate the number of cells using kind of different doses. So here you can see 3 different doses of this RNP. And here is the quantitation. And you can see that there is a dose-dependent increase in the amount of editing that you see. And then we wanted to understand what kind of cells are being edited here in the brain. And so shown here is kind of we take coronal sections and do immunohistochemistry. And what you can see here is sorry if it's really small, but what we try to understand is what kind of cell types are targeted by this gene editing. And more importantly, are there some cell types that may not be targeted? And so in this triatom, we looked at DARP32, we looked at CTIP and S100 beta. And as you can see here, or maybe not, you just have to take my word for it, is that there seems to be a lot of colocalization with DARP32 and CTIP positive cells, whereas when it's S100 beta, we didn't really see that much colocalization. It's very similar in the hippocampus. In some cases, we had expression in the hippocampus, and we saw, for instance, that there was good colocalization with CTIP2, but not always with GFAP, suggesting that maybe there is some kind of preferential tropism of this kind of a formulation for some cell types. So we haven't really investigated that, but that is something interesting to talk about. And so, of course, I set this up all as kind of a drug discovery way for Huntington's disease, right? So how would we use this as an application to Huntington's disease? And I wouldn't be saying this if we hadn't done it. So we've done some of these experiments and kind of wrapping up some of that work. But so basically, once you have the delivery sorted out, the idea would be, can you, instead of targeting TD tomato, target Huntington, the mutant Huntington gene, where you target upstream of the exon 1. So as I said, that there is expansion in the exon, there's a poly-Q expansion in the exon 1. So when you target upstream of that, you inactivate the gene. And can you make this in the form of an RNP? Can you administer it to back-HT mice like we did in the case of AZO? And can you change some of the molecular pathways as well as behavior? And of course, those are the experiments that we are doing currently. And I just thought that would give you a flavor of some of the experiments that we are doing in this area. So just want to summarize by kind of zooming out and saying, what did I talk about? I talked about kind of the aspects that are here in blue, use two different approaches, perspectives for how I think about drug discovery in neuroscience and rare diseases. There's one of them is using circuit models for developing prosocial therapies. Another is more kind of in a monogenetic space. And I think one of the biggest, something that was really interesting is that the problems are very specific for different areas. In areas where the genetic landscape is more distributed, there's very, very interesting questions about what are the pathways and targets that are downstream of perhaps a very distributed cause of the disease. Let's say for autism spectrum disorder, there's obviously some rare variants that are associated with the disease, but there's a lot of common variants, and there's been a lot of discussion in the field about maybe these common variants kind of converge on some pathways or circuits. And if one were to target these pathways or circuits that are downstream of these common variants or other causes of the disease, maybe one can kind of see changes. And this is something that I worked on during my postdoc and in my lab for a few years after that. And I think it was really interesting. We did a lot of work on discovering new targets based on these circuits, and we tried to test some of these theories. And we also did work on developing circuit models to test therapeutics, and I showed you some examples of where we applied that to work on oxytocin receptor agonists. And what I also talked about is using how do we use this in Huntington's disease, right? So using direct gene targeting and innovative platforms. And that is kind of the second aspect that I was really interested in and spent the last couple of years working on, is how can we, when you already know the target, that doesn't mean that there is no interesting work to do. There's obviously a lot of interesting work to do on how do we, even if you know the target, it's obviously not always easy. How do you target it using a therapeutic modality, and what are the challenges there? And there's a lot of interesting work that we did with Anderson's oligos and gene editing that we spoke about. And so with that, I would like to end the talk. And for my work on systems neuroscience, I'd like to thank my colleagues at Roche that I collaborated with, and for obviously the work on Huntington's disease was done in Jennifer Doudna's lab with Jen Sabo and Brett Stahl. With that, maybe I will end my talk here. And
thank you, Doctor. Thank you, Dr. Madhurima, for this very wonderful and insightful talk. We have a few questions in the chat box. The first one is, what are the tests used to measure social behavior, and is there a reason for using rats and mice?
Yeah, okay. So you mean like what I did, I'm guessing what I did in my experiment.
Yes, in the beginning, almost in the beginning.
So
Yeah.
I used three-chamber sociability test, which is basically, I think what it is kind of, it's a rectangular box. There is a central chamber where the rat is, that is kind of the starting position. And there is kind of two antechambers, and one of the antechamber has a kind of a mouse or a rat. There's some so if I'm using a rat, it has another rat which this rat has never met, of the same gender, and there is kind of it is behind a little it is in a separate room, but there is interaction. So there is place for kind of sensory and olfactory interaction that allows for sensory and olfactory interaction. That is one side. On the other side, there is an object which this test rat can interact with. So you're basically saying, are you interested in interacting with a novel object, or are you interested in interacting with a novel conspecific? And this is basically what we do. So we can measure this. If we're doing repeated tests, test, retest, what we try to normalize that to the total exploration. That's why you may have seen me doing sociability index, which means that because if you measure, if you put them in the box many times, they kind of get habituated to the box. So overall, their activity is decreased. So we normalize the activity. We normalize the social interaction to total activity. But if we have kind of only one-time testing, like we did in the chronic paradigm, we measure the number of seconds in active social interaction.
Okay. So the next question is, how long does the effect of this oxytocin intervention last?
That's a good question. So, like I mentioned, in social behavior, it's difficult to measure this over time because there is obviously this test retest. So I have noticed that even if I measure it 2 times, spaced out a couple of weeks, there is still an effect of like they get habituated. So we are not able to measure that many times, but so we don't know. In that sense, I always try to do baseline on 18 days, and we've not done after, because it's often hard to interpret this over time. But when we did physiology, right? So the effect on the circuitry, the effect of this on circuitry, even the oxytocin effect, we measured this over a 1-week window, and there was no difference like for day 18 versus like day 18 plus like a week. So if I were to hazard a guess, I would say it lasts at least a couple of weeks, but it's the drug itself is a small molecule. It kind of doesn't last very well. So I don't think that you would remodel the circuitry, but it probably lasts a week or more.
Okay. Okay. And the next question is, did the ASO therapy for HHT show any cognitive effect?
Sorry? Did the ASO therapy? Okay, yeah.
Yeah.
So we didn't measure. Actually, I don't remember if we measured cognitive behavior in package D. So usually in package D mice, we've only done rotarod, but these are not in Holly's paper. I don't know if she measured it. In the work that we've done in my lab, we've only measured rotarod behavior.
Okay.
Although there are obviously cognitive deficits in Huntington's disease patients.
Yeah. Okay. Okay. The next question is, what are the side effects of using antisense oligos, if there are any?
Yeah, I mean, this is something that's totally out of my area of expertise because, I'm doing preclinical research. So I would say that it would be presumptuous for me to comment on this. But basically, I think if you talk about the molecular level, maybe I will just talk about the molecular level. So the way you design the oligos is Watson-Crick base pairing. So if there are about 1 to 3 mismatches, there can be a biological effect on another gene. So the way we design this, we make sure that there are no other targets in the transcriptome for this antisense oligo up to 3 mismatches. And I think that's very important because if you kind of randomly degrade other mRNA, that could have off-target effects. And so certainly this is one of the design principles we incorporate while designing the drug.
Okay, adding to that, I have another question. Can we use siRNAs or any other kind of RNAs that could be naturally present also? Can they extrapolate? Can they be extrapolated in this case?
Yeah, absolutely. There is a lot of siRNA drugs now in the clinic, right? So siRNA interference certainly is something that people are trying for Huntington's disease as well. There is a few differences between siRNA interference and antisense oligo. Like for instance, I thought this is very interesting. Obviously, the antisense oligo, a lot of the effect is happening in pre-mRNA. And that's why the antisense oligos are fairly potent. And of course, there's a lot of nuclear localization of that. And I think for RNA interference, that's more cytoplasmic. And there is this kind of theory that just during mRNA processing, that if you use something to target only mRNA versus mRNA and pre-mRNA, then you have kind of higher, different amounts of kinetics and knockdown. Whereas, of course, the advantage of siRNA is the effect is amplified. So the stoichiometry of how many molecules of azo you need versus how many molecules of siRNA you need per cell is very different. So I think there's some very interesting differences that make them like azo is good for some situations and siRNA is good for some situations. So there's certainly like a place for both of these therapies. Yeah.
Okay, thank you. The next question is, how much computational modeling is needed to find the targets for pharmacological intervention that are currently useful?
Computational modeling. I guess this is about the first part to find novel target discovery, I'm guessing. Yeah. So I think a lot of the novel target discovery, there's a lot of genetic-based novel target discovery, right? And I think that there is a lot of that. But in terms of computational modeling in the systems neuroscience space, I haven't really kept up with it. Obviously, there are a lot of, blue brain connector kind of, there's a lot of really cool work that's going on. But I haven't really kept up with it. What I will say is that there are some efforts to kind of understand biologically defined subtypes of these common diseases, right? And this one can think of like if you have patients with functional MRI data, right? One can use this to understand specific kind of subtypes or kind of latent endophenotypes or kind of clusters of phenotypes that are there in this based on this data, and people are doing that in the computational space to understand, if you have like a few patients where kind of this functional MRI data, and then you have the clinical score data, there is a lot of variability, but maybe there are some subtypes that correlate with certain aspects of the clinical score. And I think that could kind of fill in this aspect of it's a very heterogeneous disease, but maybe specific aspects of the disease have specific biological basis. And that you can imagine that kind of going back and translating preclinically to say, maybe this circuitry is important for this aspect, let's say repetitive behavior. And then you could look in the circuit either to make new models or find new targets.
Okay, thank you. The next question is, what are the future plans for gene targeting technologies which will help mitigate the requirement of surgical delivery?
Yeah, that's a good question. I'm guessing that's Vidita. Yeah. Hi, Vidita. So, yeah, I think that's a very good question. So this is in the AAV space, right? There is a lot of experiments for sure in the SMA case. They're delivering AAVs intravenous, and that crosses the blood-brain barrier. And I think that is very important in this case. For Huntington's disease, also the antisense oligo is delivered intrathecally, so it doesn't need intra-ICV surgery. But for instance, if there is gene therapy or gene editing, when there is for Huntington's disease, this is going to be a big challenge of finding delivery mechanisms that can cross the blood-brain barrier and where you don't have to have local delivery. And this is certainly, as you noticed, even if you make like an RNP, it's the size of an antibody, you have to go local. So certainly there is a lot of work that's going on, can you use kind of viral vectors, but somehow have more control over them so that you can have on-off switches, for instance, or can you have other ways of crossing the blood-brain barrier and you don't have to have local delivery. But I would say this is one of the biggest challenges in the field right now.
Okay, thank you. To add into that, Pranjal has a suggestion, like what about AAV capsids? It can be modified along with some nanotech. So he's asking if that is possible.
Yeah, absolutely. I mean, Viviana's work has led to a lot of discussion in the field, right? Those PHP capsids, of course, unfortunately, they work on a mechanism that's very specific to rodents, so that doesn't translate out of the rodents. But of course, that has inspired a lot of AAV capsid discovery. I'm kind of very jealous of this field, but I'm actually not working on it. There seems to be a lot of combinatorial things that one can do to see if it can cross the blood-brain barrier. Yeah, it's a huge field of research.
Okay, thank you. I have another question. How long does it take for the entire process to be done? Like an estimate of the time that could be taken for doing this process, doing this research?
You mean like drug discovery to the clinic?
Yeah.
Okay. Yeah.
The experiments that you showed today, how long did it take for you to do?
Yeah, that's a good question. So usually, like if you're doing, it depends on the modality, right? So if you have a small molecule, this takes a really long time. Typically, I don't. So typically, the kind of my part where I'm doing like this preclinical work lasts about 5 to 7 years. Where it depends on whether you have the models, whether there's biomarkers, and so on. But in a place like Huntington's disease, where you're using an antisense oligo, there you could have much shorter development timelines, almost like 2 to 3 years, I would say. And that's mostly because of that's partly also because of where the field is. There are cellular models, there are animal models, and you can say, for instance, as you saw, we know how much lowering you need to have in an animal model to have that kind of lowering in a patient, and that is huge. If you have that in a specific disease area, we don't have that for a lot of disease areas. And so of course there is different challenges, and both of them are interesting, right? From the perspective, I mean, I like to do preclinical research, but of course we want advanced therapies. And so there's kind of these two aspects there.
Okay, thank you. There's another question. Since in pharmacological studies, type of model plays a very important role, how do we ensure the correct model, and is it possible to change the model if found incorrect at later stages?
Yeah, no, I think that's a very good question. And this is actually, I would say, if there was like one kind of common thread in kind of my talk, is this is one of the things is that the model is very important. And the only way we understand is by back translation. We often refine our models, and that is one of the things that one of the unique advantages of being in pharma is that we are able to test drugs that eventually go into the clinic, and when something translates or doesn't translate, we have the opportunity to really carefully look at the data and then say what translates, what doesn't translate, how can we improve the model. And I think that is key, because obviously it is, at the end of the day, it is a model, and one has to be very careful not to overinterpret that data.
Okay, so that's it. All the questions are done. Thank you for the wonderful talk and for answering all the questions. Thank you. Thank you for joining us.
Thank you very much. It's a pleasure.
Okay, there's another question right now. Okay, there's another question that came up right now. So what is the current status of challenges in terms of regulations when it also comes to ASO and gene editing based interventions?
Yeah, I think the regulatory challenges, I mean, I'm not very familiar with it, but because, like I said, this is kind of like, I'm so early on in the pipeline, right? And we have really cool cross-disciplinary teams at Roche, where we we work in teams where I bring something to the table, and there is someone who's an expert on regulatory interactions that is like that they have done throughout their career. And so I find myself kind of out of my wits here, but what I would say is that there is, so for antisense oligonucleotides, we really so there are already 3-4 drugs in the clinic, right? So if you're not the first drug in the clinic, we really stand on the shoulder of giants. And our partners, Ionis Therapeutics, has Ionis Pharmaceuticals, I think it's called, have already have a few antisense oligo drugs in the clinic. And so this is something that, it helps to have something that is already out there where you are able to build on that. And then for gene editing for CNS, I think this is something that is actively, an active area of discussion. If you go to any of the gene editing talks, there's always someone from regulatory talking about. There is a lot of discussion about even things like, okay, what is the gold standard for like efficacy and off-target effects? Because there is like so many ways of measuring off-target effects, like GuideSeq. There is so many things. There is no gold standard because every other day there is a new methodology that comes up. And so this is very much evolving, and this is a very active area.
Okay. Okay. To add on to that, is there any ethical concern that could be associated with this particular usage?
You mean gene editing?
Yeah, gene editing and even anti-oligo.
Yeah, in terms of like, there's a lot of ethical discussions, right, on germline editing, and so that is something. There's currently a moratorium. That's something that obviously we're not going there. We are very far from it. We are somatic editing, but also post-meiotic cells in the brain. So you just need kind of one-time delivery. So I think those, luckily, and I think this is very important to state that this is something that when you kind of come up to this point where you're introducing double-stranded breaks, that one needs to be very conservative. And so we really think that having a single-time editing in a somatic cells, and then that's it. That seems one has to kind of go in a very stepwise manner.
Okay. Okay. Thank you so much. Thank you again.
Yeah.
Thank you for the wonderful talk.